Good morning, students. So this is going to be your lecture number 8. Here we are going to talk about the resting potential, action potential, and the synaptic potential. Synapses also will be discussed along with these all topics. So in the progression of the previous after physiology, we are going to discuss about the uh, all digestive system, circulatory system. We are talking about the neuron activity. So that potential we are going to study in detail. So the recapitulation for this particular lecture is you should belong to the module number 1. Module number 1 is related to the cell membrane and its activity along with the fluid mosaic model. Uh, the whole detail about the cell membrane should be known to everybody along with the physiology of the human body, that is part of module 2, should also be known to in detail. All the physiological activities and with the structure should be known to everybody before going to understand this all. So today our objective is to talk about the action potential and the synaptic potential or synapse. So after this video you will be a little clear about the activity which is shown through this action potential and the synaptic activity. So just to go with all that, we need to understand what are neurons, and I have shown you the diagram of a neuron. You can see the diagram of a neuron that is responsible for passing the information, or you can say the synapse or the signal from one part to another part of the axon or the neuron. The excitable tissue, that neuron, is the nerve tissue, and the muscle fiber, that is having muscle tissue. Neuron is the primary structure and the functional unit of nervous tissue that is present in brain, spinal cord, that is it is passing through a whole nervous system, brain, spinal cord, nerves, and sensory cells. What are the parts of this particular? So these are part of neuron are dendrites. You can see this is soma, this is node of Ranvier, these are the axon terminals, these were Schwann cell, this myelin sheath above to these particular cells. This is axon hylux. We have already discussed every cell is covered by a particular membrane or a cell, which is also called a cell membrane. Here we can call it as myelin sheath. Axon hylux, and this is initial segment. You can see the center of this particular dendrites. This is the structure of a neuron. Now we will learn how it is going to act for a particular uh, formation, or you can the passage of information from transmission from one point to another point. So propagation of a neuronal excitation from dendrites to the axon, you can see. So this dendrites, you can see, this is the central soma part, and this axon with the axon collateral, you can see this is the propagation of uh, neuronal excitation passing from one end to another end of the neuron bilayer. So I hope you remember the phospholipid bilayer, the double layer phospholipid bilayer, the cholesterol, the glycoprotein, that is protein and carbohydrate attached together, glycolipid, lipids, with carbohydrate, and this is the peripheral membrane protein, the integral membrane protein, this is the other cytoskeletons, this is the cholesterol part, protein channel, and the phospholipid bilayer, you can see all through collectively. But why it is called as phospholipid bilayer? Already I have told you, this is one, this phosphatic composition, the another phosphatic composition, correlated to, connected to each other in the hydrophobic tail area. You can see it is also filled with the cholesterol and lipids, and because it is going to be hydrophobic, and the other part or the upper part, head part, is going to be hydrophilic in nature. So we have studied that in the cell membrane also. Now we will go to talk about the intra and extracellular ionic concentrations that is passing from one part of the cell to another part. That means we are talking about the intracellular or extracellular exchange of the ions within the phospholipid bilayer. I hope you can see this phospholipid bilayer. I have shown you here in the diagram, and you can see the exchange of chloride, sodium. Positive ions and the negative ions are going to be, you can see, This is the extracellular medium. This is the intracellular medium. So what ions are present here are going to be this is the extracellular medium, and as per the activities in the body to keep a homeostasis as well, that passage of the ion from this membrane is going through one part intracellular medium to the extracellular medium. You can see. So we can also show it like this is the sodium potassium pumping. So sodium ions and potassium ions, and inside you can see that inside the neurons only potassium ions are present. So sodium is taken externally from the extracellular medium. So you can see this is pumping sodium inside the neurons and outside the neurons. So you will find a bilayer is going to get formed, positive outside and negative inside. So inside the neurons, negative ions, you can see a layer, and other outside this is a dipole is going to develop. So sodium ion inside is around 20 mm, outside 145 mm. In the plasma, generally it is found. This is the concentration of ions found in the intracellular and extracellular compositions. So sodium ion inside is 12 mm and outside is 145 mm in the plasma. Potassium 140 mm inside, and outside it is 4 mm. Chloride, this is 4 mm inside and 115 mm outside. Bicarbonate for 12 mm inside and 30 mm outside. Protein 140 mm inside and 10 mm outside. Calcium, this is 0.0001 mm to outside it is going to up to 2 mm. So let's talk about the membrane potential. So we can see here a phospholipid bilayer. These are the outer ions and this is the inner composition of the ions. That exchange can be recorded in terms of voltage with respect to the time. So millivolt is the unit taken for voltage, and time with respect to this, it can be seconds and milliseconds. So it may be hyperpolarization, this is magnitude of the membrane, and the polarization done through the membrane, and the increased magnitude of the membrane potential can be recorded here like this. So graded local response and graded depolarization or hyperpolarization, you can be getting through that electricity, chemicals, and generator potential sensory. So the compression, deformation of nerve endings, receptors potential, and the action potential, that is synaptic activity, uh, EPSP depolarization. So the graded local responses, graded depolarizations can be captured here in the form of signals. So membrane potential, you can see this is graded potential depolarization. The lower stimuli and the stronger stimuli as a result of this depolarization, and this is called resting potential. Here we are not finding that signal or a synapse kind of thing. So initially you can, if it is a weak stimulus, you will be finding the lower kind of curving within the graded potential, and this is depolarization and this is hyperpolarization. So the kind of the curves obtained in the graded potential curves, so I have taken this for a reference of McGraw-Hill. The electric signal and the synapse, the neuron signal will be passing from one end of the dendrites to the another end of the axon. That means neuron signal passing from one to another or to the muscle or towards the gland. Electrical synapses, electric signal goes through the gap junction or bidirectional. So you can see here, this is diagrammatically I have tried to show you. This is the intracellular space, the space between the two cells. This is the plasma membrane. This is the intercellular space. The size and the thickness, it may be 2 to 4 nanometer. This is hydrophilic channel, and this is the connection monomer. You can see this is closed one and this is the open one. So how the electric signal passing from one layer of the membrane to other layer, or it is it is starting from the one point of the membrane and it is leaking through the another part. So the electrical synapse, electrical signal going through the gap junctions. So the gap of these particular fluid-like material. This is bidirectional composition can be shown here in this particular structure. So the junction at which this is con exon. It may be closed, looking like such, and it may be open, present on this plasma membrane. If I'll talk about one, this is going to be a monomer, and if many are taken here, it will be performing the activity like electrical signal in synapse. So you can see the electrical synapses, the electrical signal is passing through the gap junctions as well. This is going to be bidirectional here as well. So by diagram, we can be able to assess this. The chemical synapse, it is the chemical transmission, that means one-way transmission or one directional from presynaptic to postsynaptic cell. So it may be 1 millimeter cubic of human cerebral cortex, and about 1 billion of synapses will be captured within. So now we will discuss synapse in a detail. Like this is the axon, exodendric synapse. So you can see this is a dendron. This is one axon. This is axon terminal. And these are the dendrites of a nervous cell. This is a cell body. You can see, so dendrites are going to connect to the axon. So you can see the axon terminal. This is presynaptic activity. Axon is connected to the dendrites of the other neuron. So you can see what is present inside. So the axon or exodendric synapse, I can say. This is axon. So for that exo, and this is dendron or dendrites. This is dendric synapse. These two are going to pass the signal from one to another. So axon terminal, the presynaptic. These are the secretory gland, these are the synaptic cleft, these are the synaptic vesicles, these are the receptors, and the postsynaptic dendrites, mitochondria, the active zone here, the postsynaptic, and the membrane, activities are shown here with the connection of axon and the dendron. So the synaptic transmission can be seen here. This is the presynaptic membrane. A little larger diagram to show you better regarding the synaptic activity, how it is. This is synaptic vesicles and this is a synaptic cleft, the gap. These are the neurotransmitters and the postsynaptic membrane. So you can see the synaptic membrane, presynaptic membrane with having synaptic vesicles, and there is a gap between the synaptic cleft can be called as. These are the neurotransmitters passing from one area or one point or a gap to another. Synaptic transmission, let's talk about. This is the axon terminal, this is the cleft area, and this is the dendron spine. So you can see the synaptic transmission, it is having the voltage gated calcium channels. These are voltage gated, that means they may open once voltage is obtained here. So this is the synaptic vesicles. These are the neurotransmitters present within the synaptical vesicles. These are the neurotransmitters, and they are ready to re-uptake. And this is the whole axon terminal. So the post-synaptic density we can see here. And these are the receptor. This is the receptor area. So what is happening? So under the synaptic transmission, you will be finding that the synaptic vesicles carrying neurotransmitters on getting the voltage gated calcium channel, on getting the voltage, it may transfer the neurotransmitters to the another dendron spines. So dendrons are going to receive it and this is going to act as like a receptor. So the synaptic transmission can also be seen here can be recorded in the phospholipid bilayer. This is a ligand. These are the calcium channel. It will open ligand gated ion channel, or it may be another kind of channel can also be there. So this is ligand gated ion channel can open, and this calcium can pass from one point to another point. So the mediator molecules activate the receptors on the subsynaptic part of the postsynaptic membrane. You can see this is a phospholipid bilayer. We can see the synaptic activity where the passage of that particular can be seen. So ionotropic receptors, ligand gated channels producing either IPSP or EPSP at postsynaptic cell. So this activity can be seen here. So mediator at the extracellular domain activates the intracellular G protein and leading to the intracellular signaling. So gene expression, chemical reaction, channel opening, closing of the membrane, and permeability changes are going to be the activities performed by these particular. This is metabotropic receptors. Under metabotropic receptors, the mediator at the external domain activates the intracellular proteins and leading to the intracellular activities. So this is, you can see the glutamate is passing through this particular gate, and it is going to be the receptor finally, and the information passing from one point to another. So that you can see G protein coupled receptors can be seen. So the gene expression, chemical reaction, channel opening, and membrane closing can be found here. So summary of this particularly can be shown here through this diagram. So one exon, another dendron, the exon, you can say it is connected, and this is the synapse. This is a terminal end, and this is a synapse, you can see. So the summary, the action potential can be obtained, the voltage gated channels can be seen here. Calcium influx is also one of the activity can be found here. The vesicle exocytosis is, yes, can be shown here. Neurotransmitters are going to release these particular material, and going to diffuse onto the receptor one. So interaction with the receptor, maybe the ligand gated channel or maybe the other. So this kind of synapse activity is going to be done through. The similar kind of diagram I have tried to show you here. You can see this, the presynaptic membrane, the postsynaptic membrane, the receptor area, the ligand gated channels by which the sodium ion and the potassium ion, you can see. This is the neurotransmitters and the synaptic activity in the phospholipid bilayer membrane. The ligand gated ion channels are performing their activities in such a way. Finally, these are gated channels, and these are ion ligand gated channels. So it will close once the activity of the synapse is done. So the stimulus of the several synapses with the ligand gated ion channels can be seen. So these are the possible Q&A you can think of, and few references I have also kept here for further detailed study about the neurons, exons, and the synaptic activities. So this is all for today. Thank you.